The goal of my research project is to develop an in vitro platform for drug testing on mature pancreatic beta cells in the context of diabetes research. The most commonly used in vitro models are pancreatic cell lines, which are cultured on hard plastic. And this is lacking the physiological relevance seen in vivo. And for this specific reason, many studies are done in animal models. For example, the high-fat diet mice can develop similar symptoms as type 2 diabetic patient. For type 1 model, the STZ mice is widely used as this drug will specifically target the pancreatic beta cells and induce their death. However, these models fail when it comes to clinical trial in humans. This is mainly due to the complexity of the disease and the species-specific mechanism, which are completely different between a mouse and a human. Therefore, there is a need to develop in vitro platform with the human genetic background and physiological relevance to study beta cells in the context of diabetes and to test new therapeutic candidates. My project is addressing this exact need. First, we will improve the differentiation protocol of human-induced pluripotent stem cells into beta cells by using extracellular matrix cues. Usually, those beta cells obtained from those protocols are highly immature and need a further maturation, which is done in vivo into mice. To avoid such use of animals, we will use a platform based on the organ-on-a-chip technology that will allow us to further mature those cells fully in vitro. The Langerhans islets are not only composed of beta cells but are surrounded by many different cell types, including endothelial cells that form vessels. So as a next step, we will co-culture beta cells with endothelial cells to improve their functionality. One aspect of this novel in vitro platform is the option to run many different assays in parallel. But this requires to develop tools that can measure quickly one defined output. Here we are focusing on non-invasive tools such as Raman microspectroscopy and fluorescence lifetime imaging. And so in the last part of my project, we will combine all those different aspects to generate a drug testing platform to test mature pancreatic beta cells. This project has the potential to reduce the use of animal by replacing the in vivo maturation by a fully in vitro one, by developing human-based drug testing system for the diabetes field, and finally by contributing to the organ-on-a-chip technology field, which ultimate goal is to replace animal study by multi-organ system on a chip. I am very appreciating to have received a fund from the International Foundation for Ethical Research, and I'm looking forward to make my contribution.